Good afternoon, everyone. I will be presenting the general concept of fractional dosing of vaccines. Headlines related to vaccine shortage and the need of solutions have been common in the recent years. The first headline here from the New York Times, "In crisis, vaccines can be stretched, but not easily," talks about innovative solutions to cope with vaccine shortages, and one of these is fractional dosing of vaccines. Fractional dosing of vaccines is one of the dose-sparing solutions where Sorry, I have a problem with the passing. Okay. Okay, sorry about that. Fractional dosing of vaccines is one of the dose-sparing solutions where a lower amount of vaccine is administered to increase the number of people who can be vaccinated. This strategy is particularly considered to cope with situations of global shortages of vaccines, to balance a manufacturing process that cannot quickly adapt to varying needs, or to reduce the high cost associated with some vaccines. Fractional dosing of vaccine has been a topic of interest for several decades, and a long list of licensed vaccines have been studied over time. For the majority of these vaccines, the intradermal route has been most often studied to deliver the lower doses, with the standard or subcutaneous or intramuscular route receiving attention more recently. Doses down to 10 or 20% of the standard dose have been studied. The wide research on this area has resulted in WHO recommendations and use of fractional dosing of vaccines in specific countries or situations. This includes recommendations for intradermal administration of fractional doses of rabies vaccines and activated polio vaccines, and recommendations for subcutaneous administration of fractional doses of yellow fever vaccines. In this picture here, we see the intradermal administration of rabies vaccine, which has this use indicated in the product label. Since 2016, we have been involved in several different yellow fever vaccine studies to provide additional information to recommend fractional dosing of yellow fever vaccines for outbreak response. Current yellow fever vaccines are used since late 1930s, are considered safe and effective, and a single dose is considered to provide lifelong protection. At present, we have 4 vaccines that are qualified by WHO. In 2006, the outbreak stockpile for yellow fever vaccines was insufficient to face large concurrent outbreaks. In this situation of vaccine shortage, WHO recommended the use of fractional doses. This decision was primarily based on a dose-finding study conducted by Biomanguinhos in Brazil. Based on available evidence, that WHO recommendations were limited to certain conditions and provided preference to the vaccine produced by Biomanguinhos. To provide further evidence on the use of fractional dosing of yellow fever vaccines, 2 sets of clinical trials with associated studies were designed following a different conceptual approach. The first study was based on a pragmatic approach and aimed to have impact on the recommendations for outbreak response. Here, we included the 4 vaccines pre-qualified by WHO and aimed to assess fractional doses of currently available batches. Fractional doses were defined as 1/5 of the full dose as defined for each manufacturer. The results of the main study conducted on adults are already published and have allowed to extend the WHO policy on the use of fractional doses to all pre-qualified yellow fever vaccines. From the results from the adult study, we selected 1 vaccine to be used in the sub-study assessing fractional doses on children and HIV-positive adults. These studies have now been completed, and laboratory analysis is currently in the final stages. The second approach aims to provide information on the minimum dose requirements by identifying the minimum dose that is safe and immunogenic. In this study, we are using the yellow fever vaccine produced by Institut Pasteur de Dakar. 3 low doses have been evaluated and compared to a standard dose in adults. The first lower dose here of 1,000 international units per dose corresponds to the minimum release recommendations established by WHO. This minimum, however, is based on historical data and has never been formally evaluated. At present, we are in the process of evaluating the lowest dose that shows non-inferiority in seroconversion compared to the standard full dose. This dose will be used in the children's sub-study that will start in the coming weeks. The results of these trials could have an impact on the manufacturing process by reducing the amount of virus concentration per dose, as well as providing further insight into the results of the first trial. During the design of these trials, there were several issues that we had to take into consideration and that can serve as a lesson learned for future studies. One of the first decisions on the design of fractional dose trials is on the fraction to be studied. Different sources of information can provide an insight into the fraction that is still likely to produce an adequate protection. Dose-finding studies are generally conducted at an early phase of vaccine development and can provide valuable information on the immunogenicity and safety of different doses. Evaluation of the characteristics of the vaccine with minimum dose requirements and also batch analysis based on historical data, potency of different batches over time, can provide information on the heterogeneity of batches at release. Finally, an understanding of the production process and vaccine elements impacting the vaccine stability is necessary to establish the potential of studying lower doses. Studies assessing fractional doses might take a non-inferiority design, where the objective is to establish that fractional doses are not worse than standard doses by a specific amount. This specific amount is the potential loss in protection that we are ready to accept. A higher loss in protection can be, for instance, accepted if by fractionating doses, a larger number of people can be vaccinated, reaching herd protection. The choice of the non-inferiority margin has a big impact on sample size, but it should go beyond what is feasible to do in a clinical trial. Modeling studies can provide valuable information and help to balance the benefits of using fractional dosing with the potential loss in protection. Equivalence trials can also be conducted, but they in general require a larger sample size. Generally, the main outcome of studies assessing fractional dosing is immunogenicity. However, the exact immune parameters that are responsible for protection are not always clearly defined, resulting in a known or poorly defined correlate of protection. The laboratory assay that evaluates the immune response needs to be decided, as well as the laboratory that will perform the analysis. Vaccine immunogenicity is generally not routinely assessed outside clinical trials, and there might be few laboratories that have the needed expertise. The best time points to assess the outcome will be dictated mainly by what is known about the immune response of the specific vaccine. The assessment of the duration of protection is important, as this will impact on the need on repeated vaccination. But as in the case of yellow fever vaccines, lifelong protection data will require several decades of follow-up. It also needs to be taken into account if the immune response is different in specific subgroups of the population that will also benefit from a fractional dosing strategy. For instance, children may have a lower immune response than adults, as in the case of yellow fever vaccines. Safety outcomes also need to be considered, as reduced doses may have a different safety profile. The route of administration of fractional doses need to be decided. The intradermal route, where injection is made into the dermal layer of the skin just below the epidermis, has been the traditional route for studying lower doses. This is because it's considered to have the potential of increasing immunogenicity and efficacy. However, deeper administration into the subcutaneous tissue or muscle can also be an option for specific vaccines and can be a preferred solution. The doses to be delivered might be influenced by the injection equipment available. Auto-disabled syringes are recommended for immunizations activities because they are designed to prevent reuse by automatically blocking after a single use and becoming unusable. However, there are very few options on this type of syringes to deliver small amounts such as 0.1 ml. These generally exist to deliver BCG vaccine intradermally and are consequently of a lower length. This challenge can be overcome by changing the angle of administration, as shown in Figure 2. This can be suited to administer vaccines into the subcutaneous tissue, but maybe not suited to deliver vaccines into the muscle. The use of alternative injection devices may provide more options for fractional dosing. Finally, there are ethical issues surrounding the use of fractional doses. First, these refer to the study participants. If fractional doses results in lower or insufficient protection, study participants should have the opportunity to receive a standard dose at the end of the trial to ensure that they are protected. Second, if fractional doses show adequate results and are used outside a clinical trial, issues surrounding who gets lower doses and how these are explained to the population come up. Information on the results of the studies and the reasons to use fractional doses need to be clearly explained to the population, as well as the need of subsequent vaccination doses. There are also regulatory issues surrounding the use of fractional doses, particularly if the vaccine is not labeled for this use. As a general conclusion, fractional dosing of vaccines can be a solution for situations when a dose-sparing strategy is needed. Fractional doses need to be evaluated for each vaccine, considering the objectives of vaccination and the characteristics of the specific vaccine. As the headline from the New York Times was saying, a solution to a vaccine shortage crisis do not come easily. Not all vaccines are suited to be used as fractional dose, and studies take time. We successfully assessed the use of fractional dosing of yellow fever vaccines and contributed to a change in WHO recommendations. However, studies are still ongoing to provide further evidence. A study assessing fractional doses of a pneumococcal conjugate vaccine will start later this year. Thank you, everyone.